Welcome back to the deep dive. Have you ever stopped to think, really think, about how amazing our bodies are? Imagine if we could harness that power, to fix a damaged heart or regrow lost vision. It used to sound like something straight out of science fiction, right? But maybe the secret isn't some external magic bullet. Maybe it's actually hidden within our own cells, these tiny master builders. Today, we're diving deep into that exact idea, the incredible world of stem cells. Not just as components, but as role models guiding us towards breakthroughs in cellular reprogramming and repair. We've got a fantastic review article here from Science Journal, Stem Cells as Role Models for Reprogramming and Repair. It's packed with insights. Our mission today, for you, our listener, is to unpack how these versatile cells are shaking up medicine and what it might mean for, well, all of our futures.
Yeah, it's a fascinating shift, actually. It's not just about using stem cells anymore. The real cutting edge is about learning from them. This article does a great job highlighting how understanding nature's own repair toolkits is basically giving scientists the blueprints to engineer totally new therapies.
Okay, sounds good.
So we'll dig into the core principles, the basics, but also look at the really advanced stuff that's already showing promise in actual clinical settings.
Right. So let's start right there at the beginning. For anyone listening who hears stem cell thrown around but isn't quite sure, what is it that makes a cell a stem cell? What's the definition we're working with?
Well, fundamentally, you can boil it down to two key abilities. Think of them as superpowers, really.
Okay.
First, they have the power to differentiate. That means they can change, they can morph into different, more specialized cell types, like becoming a muscle cell or a nerve cell or a skin cell. They're the master builders, essentially.
Got it. Master builders.
Right.
And the second superpower?
The second is self-renewal. This is absolutely critical. It means they can divide and make more copies of themselves, more stem cells.
Ah.
Now, it's important to remember, not all stem cells can do this forever. Some have quite limited self-renewal, like the ones very early in an embryo. But this combination differentiation and self-renewal, that's the magic sauce.
That dual power, differentiation and self-renewal. Okay. So does that mean all stem cells are basically the same, just with these two powers? Or are there different levels?
Oh, definitely different levels. It's more like a hierarchy or maybe a family tree based on their potential, what they can become.
Okay, walk us through that. What are the main types?
Right. So at the very top, the most powerful, you have totipotent cells. These are found only in the first few cell divisions after fertilization. Think. The absolute earliest stage of an embryo.
The ultimate starting point.
Exactly. They can generate everything, not just all the cells that make up the body, but also the tissues needed to support development, like the placenta, a whole new organism, the ultimate blank slate.
Wow. Okay, so that's totipotent. What comes next?
Next down the ladder are pluripotent stem cells, or PSCs. A key example is embryonic stem cells, which come from a slightly later stage embryo, the inner cell mass of the blastocyst. Now, these are incredibly versatile. They can differentiate into any cell type found within the three main germ layers of the body: ectoderm, mesoderm, endoderm. So brain, blood, bone, muscle, you name it.
So almost everything.
Almost everything in the body, yes. But critically, they can't form the extra embryonic tissues like the placenta, so they can't create a whole organism on their own.
Okay, so they're like a general contractor who can build any part of the house, any room, but not the land or the utilities hookups needed for the whole property.
That's a great analogy, actually. Yes, they build the structure, but not the whole environment needed for it to function independently from the start.
Got it. Totipotent, pluripotent.
Well, then we have the tissue-specific stem cells, sometimes called adult stem cells or multipotent stem cells. These emerge later in development, and they stick around in our bodies throughout life, residing in specific locations or tissues.
Like the ones that repair my skin when I get a cut.
Precisely. Skin stem cells, blood stem cells in your bone marrow, muscle stem cells. Each type is more specialized. They primarily generate the cell types found in their particular tissue or organ.
So they're more like the specialist contractors, the plumber for the pipes, the electrician for the wiring.
Exactly. They're responsible for the day-to-day maintenance, turnover, and repair within their specific tissue. Absolutely vital for keeping things running smoothly.
This is fascinating. Okay, so we have these cells with varying degrees of potential. What actually controls this process, this transformation from, say, a pluripotent cell to a specific heart muscle cell? Is it all pre-programmed inside the cell, or does the environment, its surroundings, have a say?
It's definitely a conversation between the cell and its surroundings. Both are critical. The cell has its internal programming, yes, driven by specific transcription factors, proteins that turn genes on or off, guiding its lineage. But the external environment, what we call the niche, is hugely influential.
The niche. Tell me more about that. What is it?
Think of the niche as the stem cell's local neighborhood. It's the specific microenvironment surrounding the stem cell within a tissue.
Okay.
It's made up of other nearby cells, could be fibroblasts, immune cells, fat cells, you name it, plus the extracellular matrix, which is like the scaffolding the cells sit in, and various signaling molecules like growth
So
factors.
it's a busy place. Oh,
No, absolutely. And this niche is constantly sending signals to the stem cell, mechanical signals, chemical signals, telling it things like, okay, stay quiet now, or time to divide, or we need you to become this specific type of cell.
like WNT signals or BMP, those kinds of pathways.
Exactly, those kinds of signaling pathways, WNT, BMP, Notch, EGF, they're all part of this complex communication network within the niche. It controls the stem cell's behavior, whether it stays quiescent, meaning dormant, or activates to self-renew or differentiate.
And how does the cell itself respond? Does its internal state change dramatically?
Hugely. When a stem cell activates from quiescence, there are massive changes in gene expression. But even before that, the way its DNA is packaged changes. This is called chromatin remodeling.
Chromatin remodeling. Like
Well,
unwrapping the DNA so certain genes can be read.
precisely. Specific enzymes modify the histone proteins that DNA wraps around, making certain genes accessible for transcription factors to bind, while keeping others tightly packed and silent. It's a dynamic regulation that's crucial for controlling cell fate.
Wow. So it's this intricate dance between the cell's internal state, its chromatin, its transcription factors, and all these external signals from its niche.
Exactly. This tightly regulated balance is absolutely essential for maintaining healthy tissues and orchestrating effective repair when needed.
Okay, let's do a quick recap for you, our listener, just to lock this in. First, stem cells have that dual power: differentiation, becoming other cells, and self-renewal, making more stem cells. Second, there's a hierarchy of potential: therapeutic, whole organism; pluripotent, all body cells like ESCs; and tissue-specific, like adult stem cells for specific organs. Second, there's a hierarchy of potential. And third, their fate isn't just internal programming. It's heavily guided by their surrounding niche through complex signaling pathways and involves changes in things like chromatin remodeling. Which leads me to a really fundamental question. If we have these amazing repair cells built in, why aren't we just constantly healing perfectly? Why can't our bodies fix everything, like, say, a damaged spinal cord or a failing heart?
Yeah, that's the million-dollar question, isn't it? And the reality is, while some tissues are fantastic at repair, others not so much. As we mentioned, tissues like the skin, the lining of your gut, the blood system, they have robust populations of adult stem cells that handle constant turnover and repair really well. Your intestinal lining, for example, replaces itself completely every few days. It's incredible.
Right.
These stem cells often exist in that quiet or quiescent state for long periods, which probably helps protect them from accumulating mutations, but they can be rapidly activated by injury or disease.
How do they manage that rapid response without exhausting themselves?
Often through intermediate cells called transient amplifying progenitor cells, the stem cell divides, produces these progenitors, and they then divide rapidly many times to generate the large number of specialized cells needed quickly. The actual stem cell divides less frequently, preserving its long-term potential.
Okay, so that system works great for skin, blood, gut. But what about organs where we see much less natural repair, like the brain after a stroke or the heart after a heart attack?
Exactly. That's where we hit the limitations. Several factors come into play. First, some organs just don't seem to have a large, readily accessible pool of dedicated stem cells in adulthood. The heart is a classic example. Cardiomyocyte turnover is extremely low. Second, even when stem cells are present, their numbers or function can decline significantly with aging or chronic disease. They can become exhausted. Think of muscle dystrophies. The muscle stem cells are there, but they eventually get depleted trying to keep up with the constant damage.
So the repair crew just runs out of steam, essentially.
Pretty much. And then there's the brain. As we touched on, the adult mammalian brain does have neural stem cells, NSCs, but they're mostly restricted to a couple of specific niches, like near the ventricles and in the hippocampus.
And even when they do try to help after an injury, like a stroke, you said they often fail.
That's right. It's a really interesting and frustrating phenomenon. After an injury like a stroke, these NSCs do get activated, they proliferate, and their daughter cells, neuroblasts, migrate towards the damaged area. They're trying, but These migrating neuroblasts often fail to properly mature into functional neurons. They might not integrate into the existing circuits, or they simply die off.
Why? Is the environment too damaged, too hostile?
That might play a role. Certainly, the post-stroke environment is inflammatory, but a major issue seems to be one of identity. The NSCs might have the wrong patterning information for that specific location.
Meaning they make the wrong type of neuron.
Exactly. So, for instance, in the cortex, where you might lose projection neurons that connect different brain areas after a stroke, the activated NSCs migrating from the forebrain niche are often pre-programmed to become specific types of interneurons, the kind usually destined for the olfactory bulb involved in smell.
So you need projection neurons, the long-distance communicators, but you're getting local interneurons instead.
Precisely. It's like, again, sending a plumber to fix the electrical wiring. The wrong skill set for the specific repair job needed. They're not the right type of neuron to restore the lost function.
That's a crucial insight. It's not just about having stem cells, it's about having the right stem cells, making the right progeny at the right time and place.
Absolutely. And nature's system, while amazing, often falls short in these complex injury scenarios or chronic diseases.
Okay, another quick checkpoint for you listening. Adult stem cells are key for ongoing maintenance and repair in tissues like skin, gut, and blood, often using quiescent states and transient amplifying cells. But their capacity can be limited in other organs or become exhausted with age or disease, like muscle dystrophy. And critically, even when activated by injury, like NSCs after stroke, they might produce the wrong cell types for effective repair due to inherent patterning or identity issues.
And that fundamental limitation, that gap in natural repair, is exactly why the field turned towards a more radical idea, cellular reprogramming. If nature won't provide the right cells, maybe we can make them.
Okay, so if our built-in repair crews have their limits, reprogramming sounds like the plan B, or maybe plan A eventually. How exactly does this work? And how did stem cells themselves, become the inspiration, the role models for this whole idea?
Well, the inspiration came directly from observing how stem cells naturally differentiate. Scientists saw that specific internal factors, these transcription factors we talked about, could orchestrate massive changes in cell identity. The question became, could we hijack that process? Could we artificially impose a new identity on a cell?
Take control of the cell's destiny, basically.
Exactly. And the absolute landmark discovery, the one that really launched the modern era of reprogramming, was the work on induced pluripotent stem cells, or iPSCs. This came from Shinya Yamanaka's lab in 2006.
Ah, yes, the famous Yamanaka factors. I remember the buzz around that. So what did they actually do?
They found that introducing just 4 specific transcription factors, Octover, SOX2, KLF4, and c-MYC, into a fully differentiated adult cell like a skin fibroblast, could rewind its developmental clock.
Rewind it all the way back.
All the way back to a state remarkably similar to an embryonic stem cell, a pluripotent state. These reprogrammed cells were dubbed iPSCs.
So you could take a skin cell from, say, me, add these 4 factors, and turn it into a cell that could then potentially become any cell in my body.
That's the power of it, yes. It was revolutionary for a few key reasons. First, it bypassed the ethical controversy surrounding embryonic stem cells. Second, it opened the door to creating patient-specific pluripotent cells.
Meaning cells that wouldn't be rejected by the patient's immune system.
Precisely. You could take a patient's own cells, make iPSCs, differentiate those into the needed cell type, maybe neurons for Parkinson's or heart cells after a heart attack, and transplant them back without needing immunosuppression. That's the promise of autologous cell therapy.
Incredible. A personalized repair kit made from your own cells. But it sounds almost too clean. Does the cell completely forget its past life? Does it carry any baggage, any memory of being, say, a skin cell?
That's a very astute question, and the answer is sometimes, yes. It's a phenomenon called epigenetic memory.
Epigenetic memory. Like ghosts in the machine.
Kind of. Remember how we talked about chromatin remodeling, how genes are packaged? Even after reprogramming, the iPSC might retain some residual epigenetic marks, chemical tags on the DNA, or histone proteins from its original cell type.
And what does that mean practically?
It can mean the iPSC might have a slight bias. It might be a bit easier or more efficient to differentiate it back into cell types related to its origin, like skin cells from a fibroblast-derived iPSC, compared to unrelated cell types. We talk about on-end memory genes, genes that were active in the original cell struggling to fully turn off, and off memory genes, genes that were silent struggling to properly turn on.
So the reset isn't always 100% perfect.
It's often very, very good, but maybe not absolutely perfect initially. Researchers are actively working on this, developing better reprogramming protocols, sometimes adding other factors or using specific chemicals to help erase this memory more completely and improve the quality and versatility of the iPSCs. Targeting specific histone marks is a key strategy here.
Fascinating.
Yeah.
Okay, so iPSCs are about rewinding the clock back to pluripotency. But the article also mentions direct reprogramming. That sounds different. Is it like a shortcut?
It is. Direct reprogramming, sometimes called transdifferentiation, skips the pluripotent step altogether.
Dips it. How?
Instead of going all the way back to the beginning, you use a different cocktail of transcription factors to directly convert one specialized cell type into another specialized cell type.
Like turning a skin cell directly into a neuron without making an iPSC first.
Exactly. Or turning a fibroblast into a heart muscle cell, or even an astrocyte, a type of support cell in the brain, directly into a functioning neuron.
Wow. What's the advantage of doing that?
Several potential advantages. It can be faster and potentially more efficient for generating specific cell types. And because you avoid the pluripotent stage, some argue it might be safer, reducing any theoretical risk of tumor formation, although iPSC protocols are also becoming very safe.
And how do these factors work in direct reprogramming? Are they different from the Yamanaka factors?
Often, yes. They are usually factors known to be critical for the development or function of the target cell type. Sometimes they include what are called pioneer factors.
Pioneer factors.
These are special transcription factors that have the ability to bind to DNA even when it's tightly packed in that closed chromatin state. They can sort of pioneer the way, opening up the chromatin to allow other factors to come in and activate the genes needed for the new cell identity. It's a really elegant molecular strategy.
So direct reprogramming is like taking a specific shortcut on the developmental map, using these pioneer factors to force open a new path.
That's a good way to visualize it. It's a more direct conversion, pushing the cell sideways on the differentiation tree rather than going all the way back up to the pluripotent trunk and then down a new branch.
Okay, let me try to summarize this reprogramming revolution for you listening. Cellular reprogramming is basically changing a cell's identity, inspired by how stem cells work. The Yamanaka factors were key, allowing us to create induced pluripotent stem cells, iPSCs, from adult cells, a game changer for personalized medicine. But these iPSCs can sometimes retain epigenetic memory from their origin, a challenge scientists are tackling. And direct reprogramming offers an alternative, a shortcut, converting one adult cell type directly into another, using specific factors, potentially including pioneer factors, skipping the iPSC stage. This just opens up a dizzying array of possibilities. So let's talk about the payoff. Where are we actually seeing these reprogramming strategies making a difference in the clinic? What diseases are being targeted right now?
Right, the translation from the lab bench to the patient's bedside. And the progress there has been genuinely exciting, especially in the last 5 to 10 years. The number of clinical trials using stem cell derivatives, particularly from human PSCs, including iPSCs, has really surged.
What kinds of conditions are we talking about?
A really broad range. We're seeing advanced trials for diabetes, specifically type 1, aiming to replace the lost insulin-producing beta cells using PSC-derived cells. There's huge activity in neurodegenerative diseases like Parkinson's, trying to replace lost dopamine neurons, and also conditions like Huntington's and potentially even Alzheimer's down the line.
And things like epilepsy, too.
Yes, epilepsy is another target, exploring the transplantation of specific types of inhibitory interneurons derived from PSCs to try and dampen seizure activity. And of course, macular degeneration, a leading cause of blindness, is a major focus for replacing damaged retinal pigment epithelium cells using PSC derivatives.
You mentioned macular degeneration. I know vision loss is a huge area. Are those trials showing success?
There have been some very promising results. For instance, the article highlights a trial in Japan focusing on a condition called limbal stem cell deficiency, which damages the cornea. They used patient-derived iPSCs to generate sheets of corneal epithelial cells. These were then transplanted back onto the patient's eye. The follow-up, over 2 years, showed sustained engraftment of the cells and significant vision improvement in several patients. It's a really clear example of iPSC-based replacement therapy working in practice.
That's fantastic news. Real vision improvement from reprogrammed cells. Now, the article mentioned something truly unexpected, almost counterintuitive. Reprogramming cancer cells. How on earth could that work? It sounds like playing with fire.
It does sound wild, doesn't it? But it's based on a really clever immunological idea.
Oh, well?
We know cancer cells are masters of disguise, particularly when it comes to hiding from the immune system. One way they do this is by reducing the presentation of antigens, those molecular flags that tell immune cells I'm foreign or I'm abnormal.
Right, they become invisible to the T cells.
Exactly. So the radical idea is, what if you could force the cancer cells to reveal themselves? What if you could reprogram them not into harmless cells, but into cells that actively alert the immune system?
Reprogram them into immune cells.
Into potent antigen-presenting cells, specifically a type called conventional type 1 dendritic cells, or CDC1s. These cells are crucial for activating killer T cells against tumors.
So you'd use transcription factors to turn a cancer cell into the very thing that helps destroy cancer.
That's the goal. Researchers have identified factors like PU.1, IRF8, and BATF3 that can directly reprogram other cell types, like fibroblasts, into functional CDC1s. The hypothesis is that if you could deliver these factors specifically to tumor cells in vivo, you could force them to start presenting tumor antigens effectively, essentially triggering their own destruction by the patient's immune system.
Making the enemy raise the alarm against itself.
Precisely. It's still early days, mostly preclinical work, but it represents a completely novel approach to cancer immunotherapy, reprogramming the tumor itself to fight itself.
Mind-blowing. Okay, besides turning cancer cells into immune sirens, what other innovative reprogramming applications are making headway?
Another really compelling example, which is already in clinical trials, addresses a debilitating side effect of cancer treatment: damage to salivary glands from radiation therapy for head and neck cancers.
Causing severe dry mouth, right?
Exactly, xerostomia. It dramatically impacts quality of life, difficulty speaking, swallowing, increased infections. The approach here combines stem cell biology with tissue engineering. Before a patient undergoes radiation, a small biopsy of their healthy salivary gland is taken to isolate the salivary gland stem cells. These cells are then grown and expanded in the lab into tiny functional organoids, miniature 3D structures that mimic the gland.
Little mini glands in a dish.
Basically, yes. Then, after the radiation therapy is complete, these patient-specific organoids are transplanted back into the damaged salivary glands.
To repopulate and restore function.
That's the hope, and early results are promising. It's using the patient's own undamaged stem cells expanded outside the body to regenerate the tissue destroyed by therapy. A beautiful example of leveraging stem cells for repair.
That is clever. Using the patient's own backup crew. Okay, one last thing the article hints at, which sounds almost like the ultimate goal: organismal rejuvenation.
Yeah.
Are we seriously talking about using these techniques to combat aging itself?
It's definitely one of the most tantalizing and perhaps controversial frontiers. The idea stems from observations, mainly in animal models like mice so far.
What did they observe?
That transient or temporary expression of some of the Yamanaka factors, not full reprogramming, but just a brief pulse of these factors in vivo within the living animal, could have surprisingly beneficial effects on aging tissues.
Like what?
For example, this transient reprogramming has been shown to reactivate aged or exhausted muscle stem cells, improving muscle regeneration and strength in older mice. It's also been linked to improvements in other tissues and even reversal of some molecular hallmarks of aging across the organism.
So not turning old cells into young cells permanently, but giving them a temporary boost or reset. This has wider benefits.
Something like that. It suggests that maybe just nudging the cellular environment, perhaps resetting some of the epigenetic patterns associated with aging without inducing full pluripotency, could be enough to improve tissue function and potentially extend healthy lifespan or health span.
That's profound, the idea that we could potentially rejuvenate tissues systemically.
It's extremely exciting, but also requires a lot of caution. We need to fully understand the mechanisms and ensure safety before even thinking about human applications. But it shows how understanding stem cells and reprogramming is opening doors we couldn't have imagined just a few years ago.
Okay, let's wrap up this section with a final quick summary for everyone listening. We're seeing a surge in clinical trials using stem cell derivatives, especially iPSCs, targeting diseases like diabetes, Parkinson's, epilepsy, and macular degeneration, with encouraging early results. Direct reprogramming is powering innovative strategies like repairing salivary glands post-radiation and the mind-bending idea of turning cancer cells into immune-activating cells. And the truly futuristic idea of organismal rejuvenation through transient, in vivo reprogramming is emerging from animal studies, hinting at potential impacts on aging itself. Wow, what an absolutely incredible journey we've taken through this world of stem cells and reprogramming. from understanding these basic master builders to the idea of literally rewriting a cell's identity. It really feels like we're on the cusp of something huge.
It really does. And I think if you connect it all back, the core message from the science review and the field in general is about learning from nature. Stem cells aren't just tools, they're teachers. They're showing us the fundamental principles of how to build tissues, maintain health, control cell identity, and orchestrate repair. Being able to tap into and manipulate those processes, well, it's the result of decades of really dedicated basic science research finally bearing fruit.
So thinking about all this, what does it really mean for you, our listener? This deep dive, I hope, shows just how rapidly this field is moving. A future where regenerating damaged tissues or treating complex diseases using our own reprogrammed cells might shift from science fiction to clinical reality. It really makes you think. And that leads to our final thought for you today. Considering everything we've discussed, the power of stem cells, the potential of iPSCs, the elegance of direct reprogramming, even the hints of rejuvenation, if you could pick one area, one organ, one tissue, or one disease in the human body to apply these technologies to first, with the biggest impact, what would you choose and why?
That's a great question to ponder. Because the path forward isn't just about scientific possibility. It's also about priorities, ethics, and ensuring we use this incredible power wisely and safely. Where do you see the greatest promise or perhaps the biggest challenges in this cellular revolution?
Lots to think about there. Keep those ideas turning over. Thank you so much for joining us on this deep dive into the truly fascinating world of stem cells. We'll catch you next time.